All right, so in looking at prokaryotes, there's quite a huge amount of diversity, um, and a lot of this diversity has really been left uncovered by scientists because in we haven't really figured out how to grow bacteria is essentially what it comes down to. Just like species that exist in sort of the our macroscopic world, bacteria and other and archaea, um, they need specific conditions in order to survive as well. And without those, very specific conditions, they won't be able to grow and thrive. So we've kind of been looking at the tip of the iceberg in terms of prokaryotic diversity because we really haven't been able to grow that many types inside of a laboratory conditions. Um, but there is a couple of cool things at the beginning of the chapter in the very first paragraph that it says about bacteria, or prokaryotes, sorry. Most prokaryotes are microscopic, but what they lack in size, they more than make up for in numbers. Their collective biological mass is at least 10 times that of all eukaryotes. The number of prokaryotes in a single handful of fertile soil is greater than the number of people who have ever lived. So it's pretty crazy to think about. The amount of bacteria in a handful of soil, or prokaryotes, there's more of them than all of the people who have ever lived. Um, so, I mean, we've really haven't been able to even see, though, the type of diversity that exists in that handful of soil because we can't grow the bacteria. We know they're there, but we just can't figure out what they actually look like or what they actually do. Um, and so that's kind of interesting that there's a huge amount of diversity that's really left or been undiscovered, even though we've known about prokaryotes for quite a long period of time. The way that we've started to be able to figure out what kind of diversity exists in a handful of soil is by, instead of looking to grow the actual bacteria on an agar plate like you've done in your experiment, is to instead just look for the amount of different types of DNA and genes inside of that soil. And by looking at the leftover genetic contents of it, we could then determine the amount of diversity that exists there. So, as we were saying, you can't grow that many types of prokaryotes. It says that there's 4,500 that can be grown in a lab, but a handful of soil might have 10,000 different species alone. So the amount of species in a handful of soil is more than the number of species of bacteria that we can grow in a petri dish. And one of the important things about prokaryotes is that there's a huge amount of horizontal gene transfer between species. So because of conjugation, transformation, and transduction, genes are coming from all different places. And that's sort of an interesting thing to contemplate as well, is really, what sort of makes a species in the bacterial world. So what's the difference between bacteria and archaea? You notice I have real problems saying bacteria instead of prokaryotes throughout this chapter. Well, there's a few things that makes archaea kind of an intermediate between bacteria and eukaryotes. So like bacteria, archaea, they don't have a nuclear envelope. They also don't have membrane-enclosed organelles. But bacteria have peptidoglycan in their cell walls, and archaea don't, and neither do eukaryotes. Um, Another difference is that in bacteria, there's really only one type of RNA polymerase, whereas in archaea and eukaryotes, there's multiple types of RNA polymerase. Another difference is that, remember, at the beginning of protein synthesis, we always have methionine. In bacteria, it's something called formal methionine, whereas in archaea and eukarya, it's just methionine. So the difference in the type of amino acid at that first initiator sequence is one of the distinguishing markers between them. Also in archaea, we find some introns, whereas in bacteria, they're very rare. Remember, those are the non-coding sequences of genes. Archaea, just like eukaryotes, are not affected by antibiotics, but bacteria are. And in archaea we find histones in the DNA just like we do with eukaryotes. Their DNA is organized like bacteria, though, in a circular chromosome, and so you can see that they are close to bacteria, but they're also close to eukaryotes, and this is why we say that archaea are more closely related to eukaryotes, and they share an ancestral past. So where can we find archaea in high concentrations? Usually in extreme situations or in extreme environments. So archaea are what we call extremophiles, and you'll remember going back the origin of the words here, phile meaning loving, extreme obviously to the extreme of something, and so archaea we have extreme thermophiles. That means that some of them really love heat, and we find them growing, for example, in hot springs or around deep sea vents. And that's sort of something special about them. You can see here in the bottom right hand corner you've got a hot spring teeming with archaea life. And then you've also got halophiles, which are archaea that really like salt. And you can see up here in these evaporation ponds, this is where they're going to make salt, using ocean water. You can see that there's archaea, growing in high quantities here. That they're okay with high salt concentrations that would kill bacteria or eukaryotic life. There are also a special group of archaea called methanogens, and they use CO2 to oxidize hydrogen, and that releases methane as a waste product. So it's just sort of an interesting way to make a living. These would be chemoautotrophs. They are getting their energy from chemicals, and they are getting their carbon dioxide from inorganic sources. So kind of an interesting type of way to make a living. Very extreme environments. This has sort of opened our mind to the possibility that in other parts of our solar system where extreme environments exist, that there may be life. And so we talked about, on some of the moons around the gas giants, one of them being Europa. There's a lot of scientists who think there's a possibility that life could exist below in the liquid ocean. So different ways of making a living makes us think that life may not be as fickle as previously thought. It doesn't need necessarily every other or everything that we like to conceive as necessary for life, being, let's say, water and everything that we would associate with what big organisms like ourselves or cats or dogs need to survive. Alright, so prokaryotes are very important in the ecosystem, and one of their roles is as decomposers. So they take corpses and waste products and are able to metabolize them either for energy or for their carbon source, and sort of that recycles those nutrients back into the ecosystem. There are also a huge number of prokaryotic symbionts. So for example, in your intestine there's over 1,000 species, and there's actually 10 times the number of cells in your intestinal tract than there are in your body. So remember, eukaryotic cells are larger, and so just in your intestine you've got more bacteria living there than cells of your own. There have been some people who have said that really humans may just be doing the bidding of bacteria, and there have been a bunch of scientists who have tried to figure out, is there things that are, if you have a bacterial, ecosystem inside of one person's intestinal tract, does that influence their behavior? Does that influence their health? And if it does, then how much of what we are is a result of our own genes, and how much of it is a result of this symbiotic relationship with bacteria. One way of thinking is you're just a vessel for the bacteria traveling inside of your intestinal tract. All right, so most bacteria are beneficial to your intestinal tract. They help you digest things that you could not otherwise digest, and as a result, humans have the benefit of not having inflammation in our intestine and having access to some resources that we might not otherwise have. Some bacteria are parasitic, some prokaryotes are parasitic inside of us, meaning they are taking the nutrients away from us that we could otherwise have for ourselves, or they can be, what do you call it, parthenogenic and causing disease. And that's where you get a problem when the bacteria is taking nutrition away from you that you could otherwise have, and as a result you have to eat more just to get by. A large number of bacteria and other prokaryotes, they're just commensalistic, meaning that they get the benefit of living in your intestinal tract, but you see no benefit on your end, and you don't see any negative problems either. They're just hanging out there. So a lot of times the bacteria that can cause us problems are opportunistic. They're in your intestine right now, and under normal circumstances they won't cause you any problems. But there may be a certain set of environmental conditions which cause them to go into a more pathogenic, what do you call it, mode. And just an example of that could be, let's say you're taking antibiotics, it kills a huge number of bacteria in your intestinal tract, and now there's this opportunity for the bacteria that survive to grow and repopulate the area. And if the wrong type of bacteria is able to grow and repopulate the area, then you might have a problem. A lot of the bacteria that live inside of our intestinal tract release what are called exotoxins, and these are things that can cause real problems for you. We've talked about botulism before, which is when you have a can that's dented. Sometimes without knowing it, there's a little space in which these anaerobic bacteria are getting in, and you eat the food, and then you get the botulism bacteria, and then it produces this toxin, which can eventually result in death. Another example would be cholera. I think all of you have seen the movies where you've got people laying on these beds and there's a hole in the bottom of it, and they're just basically defecating endlessly water outside. So you don't necessarily die from the cholera bacteria, you die from dehydration, not being able to absorb the water that you need to survive. Sometimes the bacteria themselves are what's causing the problems or the symptoms of the disease. We call these endotoxins. An example of that is salmonella. The actual membrane itself is what causes you problems. So that's kind of another interesting thing as well, that sometimes the bacteria aren't necessarily releasing the toxins, they're a part of who they are. Bioremediation is another way that they're important in our world, prokaryotes. We've talked a lot about this before, that you can use prokaryotes to clean up environmental disasters. All right, that's it for prokaryotes. We'll move on to the protists.